Along with the times, human needs of energy has become the priority in mobilizing the daily activities. Energy becomes the important aspect of any activities, utilizing the facilities and infrastructure as the support of continuity and success. Microbial fuel cells are a new biochemical process that aims to produce electricity by using the electrons derived from the biochemical reaction catalyzed by bacteria. The energy generated by microbial fuel cells is expected to supply enough energy to partially cover the energy demands in urban. Hence, in this video, we are going to study an article written by Carlito da Costa and Hadianto, 2018, Bioelectricity Production from Microalgae-Microbial Fuel Cell Technology. In this research paper, they apply the microalgae-microbial fuel cell technology to produce bioelectricity as an alternative to a problem emerged by the use of fossil fuel usage. The use of fossil energy appears some problems, namely the reducing available volume, the difficulty of finding new reserve, the unstable price, and the pollution emerging from the fossil fuel usage. The objective of this research paper is to investigate the potential of bioelectricity production from microalgae-microbial fuel cell through the evaluated parameters of COD concentration of tapioca wastewater and the microalgae's growth rate. In the technology used, substitute energy is produced by changing the chemical energy to electrical energy through the catalytic reaction of microalgae. Light and water are used by microalgae as the energy source in conducting carbon dioxide metabolism to produce biomass, as explained in this photosynthesis reaction. The utilizing of potential combination of microalgae cultivation and tapioca wastewater to be developed, since these two ingredients are easy to find, eco-friendly, and renewable energy. The microalgae-microbial fuel cell technology utilizes the result of microalgae photosynthesis as an oxygen in the cathode with the tapioca wastewater as the anode to produce bioelectricity. Electron moving in the anode chamber is consumed by the growing microbe cells, while in the cathode chamber, microalgae convert CO2 into oxygen and biomass via photosynthesis. Oxidized mediator is transformed to the medium by the cell. The cell is repeated, and mediator is reduced by electron in catalyte. For the methods, first is the material preparation and characterization. Microalgae culture spirulina plantatis was used as the catalyte. Absorbance and the wavelength test by spectrophotometer. Meanwhile, tapioca wastewater was used as the analyte. Several tapioca wastewater characterization was also done, such as the test of sample COD content, absorbance and wavelength of bacterial cell by spectrophotometer. Secondly, the MMFC arrangement. For reactor setup, 2 pairs of reactors, each of them contain 3 liters of tapioca wastewater and 3 liters of cultivated microalgae. For membrane and graphite activation, each pair was connected by tube with membrane as the media of ion-proton transfer and as the separator between analyte and catalyte. Each reactor respectively filled by graphite electrode 1:1 and 2:2 bars. Both reactors filled by microalgae culture was aerated and 24-hour illuminated for 8 days. The illumination used 15-watt lamps with 20 cm distance. And lastly, the bioelectricity measurement. The bioelectricity was measured and tested the sample for every 24 hours. Bioelectricity observation was conducted for every 6 hours for 50 minutes for each observation. or the measurement result has shown the constant value as the optimum value. The analysis conducted by spectrophotometer, in which measure wastewater COD concentration, the density if the wastewater bacterial cell microalgal cell.
The results of this research article show some insight on bioelectricity production from microalgae-microbial fuel cell technology. The first data shows the bioelectricity production in time with representation of voltage MV against time in days for both 1:1 and 2:2 electrode. Second data shows the effect of microalgae growth in bioelectricity production of both 1:1 and 2:2 electrodes, using absorbance of algae against time in days. Third data shows the chemical oxygen demand, COD, and voltage change during microalgae-microbial fuel cell operation for both electrodes. According to the data in the graphs, this research article managed to achieve several findings on account of different number of electrodes. Firstly, on day 6, 2:2 electrode has maximum value of voltage with 166.5 MV, and the absorbance value of microalgae cell growth of 2:2 electrode is 1.5 on the same day. Second, 1:1 electrode optimum value achieved is 48 MV on day 8. The microalgae cell growth absorbance value of 1:1 electrode is 1.2 on the same day. Third, data obtained from sample analysis show that the COD or chemical oxygen demand value of tapioca wastewater is significantly decreased at 9.7 gram per liter, with microalgae absorbance value of 1.5. 4. Sample analysis result of the wastewater optical density showed decreasing absorbance value at 0.5 on the same day using 1:1 electrodes. Fifth, it should be noted that the bioelectricity obtained from microalgae-microbial fuel cell technology is small if compared to a different study. On a different research, they respectively produced 1,926 over 21.4 microwatts per square meter and 13.5 microwatts per square meter power density. The reason for this is it is due to the high carbohydrate contained in the tapioca wastewater, so it makes the hydrolysis process slower. The verdict of this research finding is it is concluded that the bioelectricity obtained has not showed significant result yet. It is due to some factors such as the tapioca wastewater is only 2 days old, and the bacteria growth is obstructed by the high concentration of cyanide shown by its high chemical oxygen demand concentration. After conducting this research, bioelectricity with maximum power density occurred on day 6, with 9.7 gram per liter COD or chemical oxygen demand concentration, 2.17 bacteria cell absorbance, and 1.5 microalgae cell absorbance on 2:2 electrodes and 0.5 inch diameter of pipes. That is all from us. Thank you and have a great day.